Hi everyone, this is a quick video explaining how to properly label cells for this job. Our team is working on a computer program that will be able to identify and track cells in a microscopy video. To do this, we've turned some of these microscopy videos into sets of images and need your help in labeling the cells. If the cells are labeled well, the computer program can learn how to label and track the cells by itself. Here is an example of the type of image you will be asked to annotate. Each panel is a snapshot in time, which progresses as you move from left to right and top to bottom. One thing to remember is that, as these images are a progression through time, cells might change position between panels. Not only that, cells may also be moving into and out of the screen, resulting in an increase and then a decrease in clarity and intensity of the cells. Because of this, you're going to have to keep track of which cells are which. In order to label the cells, you will have to color in each cell in the image, keeping colors specific to the cell. So, for example, this cell might be colored red throughout the film, while this cell might be colored green. Zoom in on the image by clicking on the magnifying glass, and then select the paintbrush tool. Choose the first color in the list on the left and color in your first cell. It's easiest to outline the cell and then color in the inside. It's important to color in the cell very carefully, coloring exactly to the cell boundary. It's easier to see if you go over the lines by adjusting the transparency like so. If you make a mistake, use the undo button here, or the eraser button here. To use the eraser, you must also unlock the color you wish to erase. You can also change the size of both the paintbrush and the eraser by using this slider here. Now go to the next panel and color in the same cell, in the same color, and continue until this cell has been colored in all of the panels in which it appears. Repeat this process for all other cells in the image. If a cell disappears by moving out of the image, like here, or dividing into two new cells, like here, don't use the color that you used for that cell for any other cell. In a similar vein, new cells should be colored with colors that haven't been used yet. Even if you think a new cell is just the return of a cell that had disappeared previously, stick to the safe side and label it with a new color. One way that cells can appear or disappear is through cell division. Cell division looks like this. A cell will become slightly peanut-shaped and then will split into two separate cells. When the cell divides, the two daughter cells should be labeled using new colors, and the color of the parent cell should not be used again. If two cells are pressed into each other, like here, it is extremely important to color the cells different colors to show that they are, in fact, two separate cells. Finally, we have adjusted the contrast on these images to make the cells clearer. However, this occasionally results in artifacts. Remember that cells are oval or bean-shaped, so any dark regions or cell-like objects, like here or here, are artifacts and should be ignored. After you have accurately colored in all the cells in the image, you are ready to submit. Good luck, and thank you for helping us with our project.